Okay, so may I just ask if you can all see my PowerPoint presentation well?
Yes, you can. Yeah.
Okay, so great. So thank you very much. I just would like to briefly introduce myself. I am Giacomo Russo, and I am a lecturer in pharmaceutical sciences at School of Applied Sciences of Edinburgh Napier University. Before we even start, I would like to thank Philippe Laurence from PIC Analytics for the kind invitation, and of course, Kate Fa from Analyza for the excellent organization of this workshop. So today, I will essentially offer a talk about the opportunities that biomimetic liquid chromatography can give in unveiling the absorption potential of new drug candidates. So let's look a little bit of the outline for today's talk. I will delve into the modeling of the drug's blood-brain barrier partitioning using a combination of biomimetic monodimensional liquid chromatography tandem UV or tandem UV MS. We will explore, albeit quickly, different selectivities in biomimetic liquid chromatography. We will look at the potential of biomimetic two-dimensional liquid chromatography, tandem UV, to model the human intestinal absorption of drug candidates. And of course, there will be time for some concluding remarks, for acknowledgement, and for some question and answers. So if you have any questions, we'll be very pleased to try to answer these either at the end of my talk or during the roundtable discussion, which will anyway follow. Okay. So the topic is essentially biomimetic liquid chromatography. What is regarded as biomimetic chromatography? So it is typically a liquid chromatography which is operated using stationary phases supporting biological constituents, for instance, membrane phospholipids or plasma protein, or alternatively, it is essentially generated by adding to the mobile phases ingredients which are able to create in solution colloidal aggregates, for instance, micelles, able to emulate the cellular leaflets of eukaryotic organism. One of the most common type of biomimetic liquid chromatography is immobilized artificial membrane, which is essentially based on phosphatidylcholine analogs, which are covalently bound to silica. Micellar liquid chromatography is instead envisaged by adding to the mobile phases micelle-forming agents, which are able essentially to create some spherical aggregates of lipid molecules, which arrange themselves in such a fashion that the apolar chain are clustered in the interior side of the system, whereas the polar heads are essentially looking at the outer environment, which is typically water. Liposomes as well are exploited in other technique, typically, but not only, capillary electrophoresis. So IAM, immobile artificial membrane, are essentially based, as I said, on phosphatidylcholine. However, to improve, to increase the stability of these phases, an endcapping is typically performed on the aminopropyl moieties. In the IAM-PC DD2, which is a marketed column by Regis Technologies, this endcapping is performed with propionic and decanoic anhydrides. It is essentially done to increase the durability of the column and to reduce possible hydrolysis problems. Essentially, what we try to do is we try to study the interaction that an analyte has with membrane phospholipids covalently bound to the silica to rationalize and extend the behavior that analytes would have with the phospholipid bilayer in vivo. The, let's say, hope and the attempt is to model the passage of therapeutics through some complex biological barriers such as the blood-brain barrier and the small intestine wall. What happens instead with micellar liquid chromatography? Essentially, the scientists add surfactant to the mobile phase at concentrations higher than the critical micelle concentration. Above this concentration, we have the formation of micelles, which act as a partition phase. Typically, we can choose from non-ionic or ionic surfactant. One of the anionic surfactant, which is most commonly used, is sodium iodecyl sulfate. CTAB is one of the cationic surfactant instead, whereas bridge L23 is a non-ionic surfactant. Sometimes it's also possible to essentially add simultaneously more than one surfactant to probably emulate more faithfully the asset of biological membranes, which are indeed made of different phospholipids or also other lipids, for instance, cholesterol. And we can essentially mimic the lamellarity and the electrostatic capabilities environment of cells. In fact, micelle essentially act as a partition phase. So during the chromatography, the analytes can interact with the stationary phase, but also interact with the micelles that are present in solution. Okay, so in the first part of this project, our aim, our scope was to model the drug's blood-brain barrier partitioning. So we all know that the blood-brain barrier is a really complex biological barrier because we have that the endothelial cell of the barrier are stick to each other by the presence of tight junction, typically claudins and occludins, hindering completely the paracellular passage pathway, but making only possible the permeation through the transcellular route, which can happen essentially both actively, so through the hydrolysis of ATP, or passively. Essentially, this is the way the blood-brain barrier look in an healthy patient, but of course, during some health diseases or, for instance, inflammation, neurodegeneration, then the leakiness of the barrier can increase and the permeability can also be increased as well. Why the blood-brain barrier is relevant? It is relevant essentially because when we look at the success rate of therapeutic targeting the different disorders, indeed, we can see that for a central nervous system disorder, we have the lowest success rate in all three phases. So it means that essentially most of the efforts are jeopardized by the complexity of the brain, yes, but also by the complexity of the blood-brain barrier, whereas the success rate are much higher for therapeutics targeting infections or cardiovascular issue or even cancer. There are different ways to sort of describe the permeation of the blood-brain barrier. One of the most historically solid and regarded is log-BB. Log-BB is essentially the logarithm of the concentration that a drug or a drug candidate realizes in the brain, divided by the concentration that the drug realizes in the blood. This is typically achieved through animal testing, so there's no other way to achieve this log-BB. And of course, the sacrifice of animals is unfortunately foreseen, so it's not really a cruelty-free way to test the capabilities of drugs to cross the blood-brain barrier. So in this first project, essentially, we tried to measure via a different in vitro biopartitioning experiment the retention of a data set of 79 compounds whose log BB were known from the literature using, on one side, immobilized artificial membrane, and on the other side, micellar liquid chromatography. But we also calculated a number of in silico descriptors through the performance of a conformation analysis in vacuum, and also trying to essentially perform some molecular docking simulation using the P-glycoprotein as a target. P-glycoprotein is one of the proteins that is more responsible of essentially pumping out the compounds from the brain. So it can be that some compounds having a high passive permeability, passive diffusion permeability, are then pumped out the brain because of the action of this protein. What did we do in terms of the method? So we tried to miniaturize as much as we could to essentially have a reduced consumption of mobile phase and also faster operational times. So for the micellar liquid chromatography, we use essentially an hydrophobic, a C18, a reversed phase, quite short, I would say, a quite short format with 3.5 particle size. And the micelles were generated by adding 50 millimolar soluble sulfate and essentially performing an isocratic elution. Because when you do micellar liquid chromatography, you don't need necessarily to actually add an organic modifier, or it's even worse because essentially it actually increases the CMC, but you can essentially achieve elution using fully aqueous media. We tried as well to apply some conditions, some isosmotic condition that could mirror quite faithfully the conditions of the physiological environment, so 9.20 grams per liter of sodium chloride, pH 7.4. For the IAM, again, we use a really short format. An IAM fast mini screening is only 1 centimeter long for 10 micromolar particle size, which is quite big according to today's standards, but it is what is nowadays commercially available. For the experimental conditions, we use to big cophosphate buffer saline, which is a buffer that is typically used, for instance, in some cellular assays, 30% methanol. Again, we applied essentially iso-osmotic condition and a pH really similar to that of the physiological environment. For the in silico properties, we essentially derived and calculated the molecular descriptors, assuming the molecules as static or also exploring the conformational space. In either cases, we consider the molecule as neutral, as completely ionized, and then we performed as a third attempt a weighted average according to the analytes pKa collected from the literature. For the molecular docking simulation, we used a 3.4 angstrom resolution P-glycoprotein, and we essentially used simulation boxes of different sizes, resembling the location in which the complexes with the ligand essentially took place. And we ran 100 runs for each simulation using the Lamarckian genetic algorithm. So essentially, we pool these affinities all together, also with the other descriptors, to see if there was any involvement of this phenomena in the passive diffusion for this data set. Okay, let's look at some of the chromatogram. In MLC, as you can see, the peak shape is not great, but this is just essentially a trait of the technique. This is because the mass transfer coefficient is impacted, and this is responsible of the peak broadening. However, the retention time was reproducible in all the runs. So essentially, it's a matter that doesn't really play a role in the, in the accuracy of the measurement. Likewise, peak shape of the IM was not great as well because the format was quite short, but still we managed to achieve some good peaks. We selected as data set 79 compounds, which were both central nervous system active and inactive, chemically diverse, small molecules only, and spanning a wide log P range, 5 units, whose log B was known from the scientific literature. Even with these regards, we tried also to consider just a few references because we realized that sometimes when you took the data from different references, also the variability of the compounds can be, of the data can be essentially really problematic. When we went to essentially metal development, we could essentially have really nice models with a squared R of 0.83 using essentially the retention indexes achieved with the micellar liquid chromatography using sodium nodacyl sulfate as a surfactant. Okay, other parameters, other descriptors that were found to be relevant in describing the passage through the blood-brain barrier were the topological polar surface area, taking into account only the nitrogen and oxygen contributions, the number of aliphatic carboxylic acid, and DDR05, which is essentially a distance deterring ring index of order 5, a topological descriptor reflecting the geometrical environments, especially in complex cyclic systems. So already we sort of managed to achieve some good models for the data set. However, from one side, the technique proved to be robust, reliable, accurate, faster, thanks to miniaturization and analytical optimization, able to, in some capacity, take into account also active transport mechanism, and by using in silico descriptor, also able to assist in some mechanism elucidation. However, the technique was not fast enough to appeal essentially pharmaceutical companies, because of course each compound had to be measured. The analytical retention of each compound had to be measured essentially individually. For this reason, what we did is we tried to enhance the industrial applicability of the technique, because essentially IAM-HPLC has always been regarded as a really good technique for studying the interaction forces involved in interplay drug phospholipid. However, the UV detection, albeit very common and inexpensive, has much lower selectivity as compared to mass spectrometry, in which, thanks to the mass charge ratio, you can essentially detect and selectively recognize the analytes also in mixtures. For this reason, the IAM-HPLC UV is regarded as a medium throughput technique, but not really as a fast throughput method. For this reason, we essentially try to transfer the method to mass spectrometry using a time-of-flight mass spectrometry machine. However, we all know, well, the analytical chemists know that this is not really a straightforward process. So you cannot just apply the condition that you have in UV necessarily to DMS for a number of reasons, specifically because the phosphate buffers that we use in LCUV to mimic the physiological microenvironment are unfortunately not really compatible with mass spectrometry, because they can seriously contaminate the ion source, thus reducing and suppressing the intensity of the signal. Likewise, typically the flow rates which are applied in LCUV sometimes are not essentially compatible with mass spectrometry, because what we are doing using higher flow rates, we are sending to DMS a greater volume of mobile phase, which is typically aqueous, because we want to study phenomena happening in water, but then this volume has to be evaporated to give the rise to the formation of ions, which is not that straightforward for this mass spectrophotometer to achieve. For this reason, we transitioned to the ammonium acetate buffer. We typically used very low concentration, 10 millimolar, instead of the phosphate buffer saline. However, this in a way pose a question: am I going to have the same analytical retention using a buffer which is not meant to necessarily mimic the biological environment? So we are going to answer this today. Well, in terms of the chromatography, we essentially managed to measure mixtures of different compounds of 7, 8. Of course, the peak shape was not great. We realized that. But then again, we are interested in retention time, and the retention time was reproducible. So it was possible, okay? But then the question here is, are those indices able, as the ones achieved by using phosphate, able to reflect, to mirror the complexity of what happens in vivo? So we first of all compared the indexes. So in this case, we could see that a highly significant relationship was achieved by essentially plotting the measurement on the IAM using Dulbach phosphate buffer, and that using the IAM in mass-friendly condition for all points but phenylbutazone and norfloxacin. This is essentially also verified in the Blunt-Allman plot that is shown in the bottom of the slides, in which we could see that the deviation, so the Blunt-Allman plot essentially showed the dispersion of the measurement for the average of the IAM retention values in both the conditions, so PBS and ammonium acetate. Essentially, the deviation happens in the middle of the range, so it's not something that is related necessarily to poorly retention or high retention. So in a way that we couldn't identify a trend in this, really. Okay, then we tried to use ammonium acetate buffers, ammonium acetate IAM indexes, and Dulbecco phosphate buffer saline indexes in modeling the blood-brain barrier passage using log BB as validation value. And what we essentially saw is that the accuracy of the module was essentially the same. So you can see that the squared R of 0.78 was observed in both IAM based on ammonium acetate and IAM based on Dulbecco phosphate buffer saline. So this means that essentially I'm not paying a huge price in terms of compromise using buffering agents which are different from those I will find in a physiological environment, which was a good news. But let's look at the throughput now point of view, because we did all this essentially to increase the throughput of the technique. Well, we managed to screen our data set, so nearly 80 compounds in triplicate in only 1.1 day, okay? Which is way less than the previous work I've shown you before, in which 6.1 days were needed. These two were anyway much lower than the other times reported in the literature applying the same technique. If you are interested in the references, you are very welcome to go and check the relative publication. So in terms of time, it was a much faster approach for our purposes. Let's look now at the effluent consumption and eco-sustainability, because of course, now we are actually really focused on respecting the environment and to design solutions that are eco-sustainable. So to screen the entire data set in triplicate, we essentially used 500 ml of mobile phase, of which essentially 400 ml of water and 100 ml of methanol. So if you compare the eluent consumption, this is really good as compared to the other methods that are reported in the literature. Clearly, micellar liquid chromatography, being based on only water, essentially don't use any methanolic fraction, or you can essentially use an organic modifier, but this is not really sort of advisable for a number of reasons. Okay, but then we wanted to go further. Okay, so essentially the blood-brain barrier is not made only by phosphatidylcholine, right? So we have plenty of lipids. And one of the most interesting is sphingomyelin, because sphingomyelin is a really fascinating molecule. It segregates in lipid rafts along with cholesterol, which are involved in signaling. But there are studies which actually demonstrate that sphingomyelin is also present and concentrated in the outer membrane leaflet of biological membranes. So it is one of the lipids that most readily interact with the drugs, okay, that approach the outer leaflet of the membrane. For this reason, we characterize the selectivity thanks to a joint activity, joint collaboration with Giulia Carone and Giuseppe Mondi's group from the University of Turin. We characterized the selectivity of a novel IAM phase based on sphingomyelin, which was synthesized as a prototype by the separation science group, where I was working essentially as a postdoc. So sphingomyelin is present in a concentration from 10 to 20% in all the body districts, but in the blood-brain barrier, actually sphingomyelin concentration rises up to 35%. So it is essentially the lipid that is most represented in the blood-brain barrier. So we want really to study the selectivity of this stationary phase. So first of all, what we did, we measured the retention of a pool of neutral bases and acids, and we could see that a good relationship was visible between the retention of neutral compound and their lipophilicity measured in the normal octanol water partitioning system. This is really similar, identical to what happens with the IAM-PC MG and DD2. However, unlike what happens with the other IAM phases commercially available, we see that essentially in most cases, the presence of an electric charge is always disrupting the interaction, and this disruption is essentially higher for bases. So essentially bases are retained less than neutral isolipophilic compounds. This is actually a reversion of the pH piston hypothesis that was formulated by Alex Abdif, observing the partitioning in liposome, and then extended this theory to the IAM-PC phases. And this is really an interesting point. So we then used block relevance analysis for characterizing the retention of analytes on the sphingomyelin phase and compare this to normal octanol water lipophilicity and also retention on the IMPC DD2 phase. I won't go that deep into the block relevance analysis plot because Laurence actually yesterday already described a little bit what each block is representing. It is essentially representing a peculiar interaction pattern, okay? So it was very interesting to see that the retention, so the retention pattern, according to the block relevance analysis, was very similar, but not identical for the IAM SPH and the IAM PC. The polarity contribution in both cases was lower than the one you will see in the normal octanol water lipophilicity. So it was also good news because apparently the role played possibly by free silanol group was negligible in both cases, which can be straightforward for a commercially available column, but not really for a column that is essentially synthesized in house. Okay. And then we also tried to see whether this new IAM phase was relevant in studying the process actually happening in the blood-brain barrier passage. So essentially, we found a nice relationship between the retention on this sphingomyelin phase and unbound fraction in plasma. So we also, in this paper, sort of hypothesized or even recommended a combined use with PAMPA BBB, which can provide some usefulness in try to mirroring the fraction unbound in the brain. So PAMPA BBB is essentially a variation of the conventional PAMPA, which is done using rather than lipids in dodecane, essentially porcine brain lipid extract. So it's actually an extract that is able to mirror much more closely the interaction taking place essentially in the brain. We wanted then to measure and to try to use essentially biomimetic liquid chromatography to study the human intestinal absorption of drugs. We wanted to use, in this regards, a two-dimensional LC approach. Okay, so we all know what happens when a pharmaceutical dosage form is essentially administered. We have the disaggregation of the unit and then the dissolution. Then the molecules essentially interact with the membranes of the microvilli, and they are absorbed through this huge superficial area. But then after absorption, they interact with the plasma protein which are involved in the distribution. So there is actually an interaction between these proteins and then the compounds are distributed toward the whole body, essentially. So we have two main proteins which are essentially in charge of the distribution. One is the alpha-1 acidic glycoprotein. However, the main one is the human serum albumin or HSA. So this is essentially the most abundant plasma protein in plasma. And we know that the human serum albumin is essentially cationic, and it is able to bind, to interact preferably with neutral and essentially anionic compounds. In most cases, the interactions are aspecific and are hydrophobicity driven. So the more hydrophobic the molecule, the more it will interact with the human serum albumin. However, the human serum albumin does support two major allosteric sites, which is site 1, responsible for the binding of warfarin, and site 2, responsible for the binding of diazepam. For this reason, the human serum albumin can indeed provide some usefulness in the resolution of the racemates, specifically if those are acidic. And this, as I was actually emphasizing before, is massively involved in drug distribution. Okay, so we have that. We have absorption first and distribution later. But then we wanted to study these two phenomena in the same analytical platform. And actually, two-dimensional liquid chromatography seemed to us the way to go. So we decided to use, in the first dimension, human serum albumin, okay? So this is essentially a commercially available column by Dysel. And in the second dimension, an IAM column. We wanted to develop a platform that could provide usefulness in screening drug candidate libraries according to their potential to cross, in this case, the human intestine. Okay. We had to build up a 2DLC essentially in-house in this specific case. We use a two-position 10-port switching valve that is essentially a valve that is able to transfer the effluent from the first dimension separation directly to the second dimension. So essentially we had two pumps, one for the first dimension, which is a quaternary pump, and one for the second dimension, which is instead a binary pump, and essentially two UV detectors, okay? And we can use essentially a one dimension column for the first separation and a second dimension column for the second separation. If we look at the methods, essentially for the first dimension, we applied a linear gradient, I would say. So for the first 20 minutes, it was just an isocratic elution, but then linear gradient, and then we kept the higher allotropic strength condition for essentially another hour. We used 2-propanol as an organic modifier, and the temperature was set at 30 Celsius degrees. So again, we installed a 100 microliter loop, because if the flow rate is 50 microliter per minute and the modulation time is 2 minutes, essentially, we were able to transfer all the fraction entirely to the second dimension. In this case, what we have a comprehensive biomimetic 2DLC setup, because there is no fraction that is not transferred in second dimension. So I cannot lose any peak. In the second dimension, it's very challenging actually to sort of develop a method, because you have a really short time, only 2 minutes, for eluting all the compounds. So we did different conditions. We did a normal gradient and then a mixed gradient, but essentially what we did here is using a lower allotropic strength condition from 0 to 36 minutes, and then from 37 to the end of the run, we sort of used higher allotropic strength condition to sort of elute the more lipophilic compounds. We used a normal, let's say, IAM-PCDD2 of 15 centimeter length, so a really common 4.6 millimeter internal diameter, so a really common format, I would say. How can we validate this technique? So we had the log-BB before, but here we need some data that can be used to sort of find out if we are able to develop really a biomimetic platform. We decided to go for the Loci-GUT gold standard technique for assessment of the permeability through the intestinal wall. So it's a really, really, really invasive technique in which human healthy volunteers are anesthetized, and essentially 2 inflatable balloons are placed into the distal segment of the intestine and then inflated. You have a multi-channel tube in which you essentially let flow a solution loaded with a known concentration of the drug you want to study the permeation of. And then you collect the perfusate at the end of the multi-channel tube, and you essentially study through an analytical technique how much has been lost during this path, and you assume that that amount has been absorbed, okay? Obviously, this technique is not for every day because it's extremely expensive if performed on a regular basis, but it's also raising a lot of ethical issues. There are drugs having really serious side effects, you know, antitumoral drugs, antitumoral agents, etc. And there are lots of paperwork connected. So if, you know, we or someone else found a way to sort of surrogate those indexes, that would be advantageous for sure, because it's really lengthy as well to do. So this is essentially the 2D LC chromatograms that we get with the whole mixtures of compound that we manage to screen. As you can see here, the separation was quite good. So we essentially exploited most of the chromatographic space. We did have the separation of some acidic racemates, for instance, ketoprofen, fluvastatin, fexofenadine, but also some actually basic verapamilase basic compound as well. But now what we want to know, is this going to give us, can we extract some sort of information out of this chromatogram. So if we just select the compounds having higher intestinal effective permeability, we could see that most of the compound essentially clustered in this space, but these two, which were found to be active transport substrate. Essentially, IAM cannot really account for active transport mechanism, essentially because it happens through a protein channel, which is not really represented by the structure of the stationary phase. But then what is also interesting is that if we look at some literature from by Claravalco, that's here, and co-workers, we know that we can combine IAM and human serum albumin to gain essentially information about the volume of distribution, but even more about the percentage of plasma protein binding. So there is the potential even to translate the retention time in the first dimension, essentially from analytical measure in a biopharmaceutical measure, so in the percentages of plasma protein binding. And so it is potentially even possible to pool the compounds according to their likelihood to bind plasma protein. Okay, so I've come to actually the end of my talk. So to sum up this last part, we developed an unprecedented 2D biomimetic comprehensive platform, which was also validated according to its potential to deflect to some capacity in intestinal absorption of drugs. What is advantages here is that essentially you don't need to run any sort of complex math or algorithm, because it essentially studying the place where the compounds elute according to this technique. And we could also have some nice analytical outcome, for instance, chiral separation and the assessment of plasma protein binding. I would like essentially to acknowledge, because of course this is a team effort, I'm just the one who speaks today, but it's essentially a team effort. So Frederick Linen, head of the separation science group from the University of Ghent, Belgium, Giulia Caron and Giuseppe Permondi from the Department of Molecular Biotechnology and Health Sciences, Lucia Dugrumetto from the Pharm Analysis and Biopharm Laboratory of the University of Naples, Federico II, but also so many other experts that influenced me deeply. Laurence Philippe, Gilles Goetz, Roman Sux from Pfizer, Claravalco, of course, Matthijs Bert, Ardiana Kaitazzi, Dieter Werzele, Ravindra Suryakantegad, and Ilaria Neri. And of course, thank you all for your attention.
Giacomo, thank you so much for sharing all this experiment. It's fascinating, and also it goes with what we said about yesterday about how we could use chromatography to really kind of highlight, so in vitro and in vivo parameters, and I think I love it. So thank you so much. Just for me, a quick question. Do you plan from this 2D platform, do you plan to build a model and kind of prediction tools based on that, or is it still at the experimental data right now? And
we are currently working in different ways. So one of the ways is actually to sort of extend the data set, because this was at the beginning more like a proof of principle. So we use around 30 compounds. So we plan to essentially expand the data set. The main problem related to that is that, as I said before, loci gut technique is hardly performed, so there are not so much data using this specific technique, but most is being published. So we hope to gather some data from that. And we are also trying essentially to exploit other selectivities in the different dimensions. So for instance, the sphingomyelin instead of the DD2, and also try to also use other stationary phases as well to try to see if the analytical attention sort of give us additional information.
Yeah, it's really good. I think Clara, do you want to ask your questions?
Yes, I can ask. So thank you very much for inviting me for this really lovely workshop, and thank you very much, Giacomo. This was a beautiful presentation. I envy you for these videos. So attractive, and I could use it for teaching as well at the UCL. So I will ask help in that. So my question was, and I have even more questions since then, that when you compared the phosphate buffer saline and the ammonium acetate, was it the same, exactly the same column, IM column, or you use different?
Yes, it was the same fast mini screening column, the shorter format. It is essentially very similar to a column guard, but it's indeed a column, or at least Regis Technologies sells this as a column. Yeah, it's exactly the same column.
Yes, because sometimes, you know, one IM column to the other can show a little deviation. I was a bit surprised that I found it a bit too big, the deviation between them. And in both cases, you used methanol and isocratic.
Yeah. 70-30, 70 buffer, which was Dulbecco in one case and ammonium acetate in the other case, and methanol as an organic modifier, 30%. Yeah, it was exactly the same. It was exactly the same piece of hardware. So
how can you explain those two outliers?
I cannot.
Some ion pair formation with the ammonia. I don't know. I found it too big, because, as you know, we always used ammonium acetate. And by the way, we always use acetonitrile because we found the peaks are much nicer with the acetonitrile and the gradient. So it's quite different. And it was...
yeah. Sorry, go ahead.
Yeah, so I was a bit surprised of the variation, and I am curious what would be the explanation. My other question is this two-dimensional. I didn't really get exactly the platform. So you elute from the HSA column and you inject the eluent to the IAM?
Essentially. Yeah, essentially you can have two approaches. You can have an offline or you can have an online. So offline means that you are collecting the fraction and then later on injecting in the other dimension, but this is not what we did. This was an online. So essentially in that 2 minutes, you collect 50 microliters per minute, so 100 microliter, and then the valve will just transfer that amount in the second dimension, and then you have the separation of that fraction. And then you pull together the analytical signals in the 1D and the second D, and then you can really see the peaks, how the peaks sort of spread in the plot. Yeah,
but if you have the pure component, you can inject separately, no? You don't need to collect if you know what you are looking for.
Yeah, it's more like that you want to study the separation dynamically in both the conditions. So what I did, because it was UV, so I injected the compound individually, and then, you know, as a proof of concept, I injected all the compounds in a mixture, you know, but yeah.
Yeah, because I don't know, you know, I at the moment, I can't really figure out what is the advantage of your plot, or just using the IM retention and the HSA retention and plotting the two together.
Well, it's just, well, of course, the data, the way data essentially will look like is very similar, but actually the fact that you can run a separation using different selectivities, that you can easily, for instance, change the method online and so see really the impact that, for instance, different ingredients, organic modifier have on the separation in an online condition. And essentially, you were right. So if the compounds are, since the complexity of the sample is also increasing, having essentially two selectivities at the same time can also support us in essentially getting a more orthogonal separation as well. So a better separation at the end of the day. Yeah.
And did it show good correlation with this intestinal absorption data?
Well, actually, we did more in the paper. We did also correlation in 3D plots with log P, with log D. Essentially, there was a cubic box in which all the compounds having higher chance to cross the intestine sort of clustered. But as I said, these are not so many compounds. So of course, further studies are needed. It was more like a proof of concept. And we are keeping on doing this because in the meantime, I moved to a new institution and then we also got a new 2DLC. So we are keep doing this here at Napier as well.
Yeah. I was also impressed with the new type of IAM phase, the sphingomyelin phase. I saw the publication. Do you think it will be commercially available soon?
Well, actually, this is a prototype that was designed by Dieter Werzele and Frederick Lienen. I don't know, honestly, if there are plans to sort of, patent or to sort of look at commercial exploitation. But I hope so, because it's anyway a bit different. I think it sort of conveys information that are a bit different than the ones that you can sort of extract from the IAM-PC phases that are commercially available.
Yeah, I know. I can see Shenaz and the GSK colleagues are here, and they are very keen on trying also other phospholipid phases, especially the respiratory, the phosphatidylserine, which is more
the negatively charged,
in the lung, and they are working on respiratory. Yeah, it's very, very exciting, and it's so nice to see the potential of chromatography, which is such a powerful separation technique. And I think analytical scientists have a good feel of the compounds, really, the properties, but can be useful for drug discovery. So if we can separate even enantiomers, just like our body, so we can catch the compound property very accurately and with high resolution with chromatography. It's great to see the advances in this field.
Thank you, Clara, for advocating for analytical chemists. I really love it.
So yeah, I am an analytical chemist.
Yeah, I know.
And I love chromatography. You see, I retired, I booked immediately an HPSC. It just makes stress relief when I can listen.
Yeah, thank you so much. If you don't mind, so just for the sake of time, we just have one last question before we move to Rob. Rob's presentation. There is a question about for you, Giacomo. How did you use the computational work to support this research?
So essentially, what I do, I prepare, a spreadsheet, containing the different in silico properties that I calculated, and then I just consider the properties achieved experimentally. So in terms of the P-glycoprotein, is essentially the free energy, the affinity for that binding site that I used. And then I essentially run a script that will will rank the descriptors according to the relevance in modeling, in sort of describing the process.
Okay, yeah. So you use not a specific computational tool, but you use what was available. I would say more in terms of regular.
Yeah, well, actually, for the modeling itself, I use Vega ZZ, which is a really great piece of software by Dr. Vistoli and Pedretti from the University of Milan. And for the other descriptor, I use Dragon, which is by the Todeschini group.
Yeah. Okay. Sounds great. Thank you so much.
Thank you.